Hello, everyone, and welcome back to Psychedelic Vantage. Today, Adam and I have the pleasure of speaking with Sam Banister, Chief Scientific Officer and co-founder of PSYLO. PSYLO is a biotech company developing next-generation therapeutics inspired by psychedelics from nature. Using computationally enhanced drug discovery, the company is optimizing the pharmacology of their next-generation compounds to ensure that their benefits reach a wider patient population. Sam, thank you so much for coming on today. How's everything going?
Yeah. Thanks so much for having me on, Jake. Yeah, things are good. I was just saying to Adam, I wrapped a grant submission today for an NIH grant, so that's a pretty good way to end the week. It's been a big few weeks.
Awesome. That's awesome. So we'll get right into it here and talk about, Silo's pipeline. And actually, before I get started on that, can you give the audience maybe some background information about yourself and how Silo started?
Yeah, sure. So I'm a medicinal chemist by training, and I got my PhD in Australia, and then that work took me to Stanford University for a postdoc. And a lot of the work that I was doing was focused on the development of new radio tracers, so things that traces that can image things like brain inflammation using a technology called positron emission tomography, PET imaging. I did a lot of structure-based drug design there, looking at sort of functionally selective new cannabinoid ligands for pain and other conditions. And I was involved in spinning out a company called Tranquis Therapeutics out of Ed Engelman's lab. That was focused on a new signaling pathway that's relevant to all sorts of neurodegenerative diseases. So we were developing drugs for ALS and for other things. So yeah, my interests broadly are in drug development. How can we develop medicines that improve lives for patients? And most of the work that I've done has been focused on diseases of the brain, generally, I would say. So I was wrapping up an academic position in Sydney, heading up some drug discovery for a philanthropic initiative there that was looking at new epilepsy treatments based on molecules found in cannabis. And I met Josh Isman around that time. And his wife was running a commercialization center at the University of Sydney. He's an American, actually. He's from New York. He's got a lot of New York hustle, and we just kind of hit it off. He was reading Michael Pollan. I'd read some of his books prior, and we just got discussing all this potential that psychedelics were showing in a number of psychiatric indications, things like depression, and also discussing some of the issues around sort of broad deployment of psychedelic assisted psychotherapy as it's being trialed at the moment. So yeah, that was really the impetus for starting the company. I had a pretty good network through the psychedelics community in terms of researchers, quite a bit of experience working on the development of central nervous system drugs, and we just started thinking about how to build better molecules for patients.
All right, so when we bring up the pipeline of Silo, something that really stood out to me when I first looked at this company is just the variety of different types of drugs that they're looking at in terms of how they relate to psychedelics. So, very cool to see non-hallucinogenic, hallucinogenic, and also less hallucinogenic, and really excited to ask about kind of all three of these families of novel drugs. So I'd like to start off with asking about, Silo 100X series, non-hallucinogenic. So some viewers may be familiar with companies like Delix Therapeutics, Gilgamesh Pharmaceuticals, some of the, you know, the well-known names of these of companies looking at non-hallucinogenic. And with Delix Therapeutics, I did some reading. They're looking at something that could be, a pill that's taken on a monthly basis, even. And meanwhile, I heard Jonathan Sporn from Gilgamesh say how they're taking more of like a microdosing approach every maybe 2 to 3 days. And with their drug also, it has like a ceiling effect to where no matter how much someone would take of it, there would be no hallucination. So I would like to ask you, is Silo what type of dosing regimen would they be looking for this series? And if you have, are you able to share the indications that you're at least thinking about at the current moment?
Yeah, sure, Jake. So yeah, PSYLO 100X is our most advanced candidate. So it comes from a sort of series of related compounds, all of which are characterized as being non-hallucinogenic, 5-HT2A preferring agonists, so some molecules that activate that receptor. we're particularly interested in psychiatric indications because that's where most of the preclinical data is to support this sort of profile for a drug. But we're open to other applications as well. So a lot of the sort of preclinical behavioral work that we've done in various rat and mouse models of behavior paradigms that are thought to be related to human disease, aspects of depression or anxiety, are all focused around psychiatric indications.
Thank you. And can you also elaborate on the dosing regimen that PSYLO would be looking at with this series?
Yeah, that's a really excellent question. For a lot of these compounds that others are developing that are described as being sort of non-hallucinogenic, there's some uncertainty there around how frequently these compounds would need to be dosed. So with a lot of therapeutics, you're typically trying to reach what's called a steady state concentration, some kind of fairly consistent level of the drug to have a therapeutic effect. That's pretty typical for a lot of psychiatric medicines, but also other drugs. What's really interesting about a lot of these compounds is that from the preclinical data we have, so sort of in vitro cellular data and some of this animal data that I mentioned, one thing that's very unusual about activation of 5-HT2A receptors is that a lot of these effects appear to happen very quickly. So they're almost immediate following some kind of acute dosing, and they seem to be enduring. So we see some of the behavioral properties of these compounds persisting, many days after they've been dosed, sometimes even longer in some of the papers that have been published from Dave Olson's lab and elsewhere. So that's a little bit open-ended for us. We have all the usual considerations around scaling from rodent models and others into humans. So using an approach called allometric scaling, thinking about how to scale dosing based on different metabolisms across different species. But an additional complication for this particular type of compound is that, we just don't know how frequently we're going to need to dose these things to get those sort of persistent therapeutic effects. It's looking like it's very unlikely these things will need to be taken multiple times a day. It's very likely to be a sort of once daily medicine or even less frequent than that.
While we're talking about the non-hallucinogenic series, I did read an update from an article, I believe that PSYLO entered into a research agreement with a very large Japanese pharmaceutical company, Daiichi Sankyo, and it talked about how the research agreement will be looking at non-hallucinogenic. Are you allowed to share which series? Is it PSYLO 100X, PSYLO 500X, or can't touch on that at the moment?
Yeah, we've got this sponsored research agreement with Daiichi Sankyo, which we're hugely excited about. Daiichi are also very excited about it. We've had a lot of great meetings already with both people in the sort of business development roles within the company, as well as a lot of their neuroscientists and other researchers. I would say their interests are not so much in a specific chemical class or candidate, but as in questions around the sort of underlying mechanisms of how these non-hallucinogenic drugs are having therapeutic benefits. So yeah, there's not a whole lot I can say for reasons of confidentiality, but I would say that Daiichi is very interested in sort of understanding how the therapeutic benefits are derived from this sort of class of molecules generally.
Yeah, that was very interesting to see that research agreement. And, Adam and I have always ourselves wondered why it's the large Japanese pharmaceutical companies specifically who have seemed to really show interest in the psychedelic medicine space.
Yeah, that's
with it, yeah.
that's a point I'd noted as well. And I'd say we have had communications with other companies historically and more recently, and they generally typically tend to be I would say from East Asia, Japan and Korea as well, which is sort of interesting because, traditionally, cultures in East Asia are not very open to these, to psychoactive substances generally, if you look at policy and law in those countries. And I think this is why there's such an interest around non-hallucinogenic compounds, so things that are having the same sort of therapeutic properties but are not psychoactive in any way. And that shouldn't be that surprising because Japan has a very strong tradition around sort of neuroscience research and drug development in the neuro arena. So it's not especially exciting, not especially surprising, given that historically, you've had some very big companies like Takeda have really prominent neuropsychiatric research departments.
This question actually really excites me that we're going to get to here with PSYLO 300X series, hallucinogenic. So, the first thing in my mind when I saw it, I was like, okay, it's hallucinogenic, but, it doesn't say if it's inspired by, NN-DMT, psilocybin, 5-MeO. So what I'd like to ask you is for PSYLO 300X, are you looking for a shorter trip duration? Can you give us any detail at the current moment as to what type of, you know, trip experience PSYLO is aiming for with PSYLO 300X?
Yeah, so PSYLO 300X is largely a tryptamine-derived series of compounds. It's actually how we got started with the company, thinking about the need for shorter-acting agents. So in Switzerland, you'll notice a lot of the clinical trials tend to use LSD because there's sort of a historical precedent with the compound being discovered there, and historical use of that compound clinically. There's a bit of a preference there for LSD, but in other parts of the world, in a lot of these trials, people are using psilocybin. Compass is obviously very advanced, and Usona as well with their psilocybin trials. But even psilocybin has this, fairly lengthy duration of action, 6 to 8 hours. You have compounds like 4-hydroxy-DiPT that Shulgin developed. Field Trip was looking at developing this as a replacement for psilocybin. But our thinking was, which aspects of the tryptamine skeleton could we modify in a rational way to make them more susceptible to human metabolism, so that we get a compound that is essentially the same sort of subjective profile as psilocybin, but lasts for a much shorter duration of action. So can we use modern sort of medicinal chemistry techniques to rationally engineer a compound like that? It's very challenging. It's not as straightforward as people would think to get a compound's duration significantly shorter. Gilgamesh is focused on an IV route of administration, so intravenous delivery of a tryptamine for a sort of related compound that they're developing. But we're pretty firmly focused on an oral tablet because that's what's going to be most acceptable for people. So that whole series is really focused around shorter acting tryptamines, intended to be very similar in the clinic to psilocybin in terms of their efficacy, but with a much shorter duration of action. And as we were working on that series, we started, we were a heavily chemistry-centric company when we started. And we were really focused on exploring interesting areas of chemical space that others weren't looking at. So obviously around the tryptamine scaffold, a lot of different companies were doing a lot of work. There's a huge amount of prior art there. And there's probably also a limit to how much really innovative development you can do. So we started looking beyond the tryptamine scaffold, and that's where we discovered all these other really interesting new bits of chemical matter for development.
I want to chime in a second here. Is there like an optimal ratio in regards to the duration to the durability of the effects that you guys are looking at?
It's really just a matter of practicality. So it'd be very pragmatic to have something that's on about the same time course as ketamine or thereabouts. Certainly a compound that can be dosed and have a patient recovered within 2 hours, say. But that might be very hard to achieve in practice. So there's no ratio, because generally what we've seen from a lot of other trials is that the sort of therapeutic effects, all things considered, between psilocybin and LSD when administered as adjunctive to psychotherapy are actually pretty similar. So it doesn't seem to be so much the duration of the drug. It seems to be a lot more about the subjective experience, the sort of intensity of that experience in terms of symptom reduction in things like depression and anxiety. So my belief is that the duration probably doesn't have much of an impact, but the subjective quality of the experience is important. So if we can make that as short as possible, but as similar as possible to psilocybin or LSD, then you have an ideal drug. Obviously, people are looking at things like DMT as well, and 5-MeO DMT. These have quite a different profile, I would say, to psilocybin. There are challenges with administration there as well. So, DMT is not going to be an orally active drug. Neither is 5-MeO DMT. So there's challenges there in terms of how you're actually going to deliver these things to patients, and whether patients will be okay with an inhalable or an inhalable product or an injection or something else.
Also add talking about the short duration and how that could have an impact on durability. Sam, I'm sure you saw the results too. I know I'm sure many of our viewers, when Small Pharma put out that data on NN-DMT, we saw, people with major depressive disorder, many of them still in remission 6 months later after one dose. So Adam and I have spoken a lot about this. It seems like with psilocybin, you really, and this is just speculation on my part, it seems like with psilocybin, you maybe get to what's called, I guess, the root causes of your trauma. Maybe you look back at different moments in your life and stuff like that. But it seems with, NN-DMT and 5-MeO, it's just speculating that the mystical experience itself might be a big driving factor. I'm sure there obviously you pay attention to I guess the debate between non-hallucinogenic and hallucinogenic, so maybe it's really just about the neuroplasticity, and we can maybe touch on that a bit. But yeah, just thought I would add that in there, and it's very interesting to follow all these trials.
There's some great data there showing that for end-of-life anxiety, so anxiety associated with terminal illness, typically cancer, as well as some of the depression trials, that the reduction in symptoms and the durability of those effects does seem to be correlated with both across a number of different measures, including dose, subjective sort of intensity of the experience overall based on a number of questionnaires, and the sort of mystical type nature of the experience based on the sort of corresponding survey there. So that clearly does seem to be important for at least some of these conditions and some of these drugs as they're being used. And I think this debate around the underlying sort of biochemical changes that are happening, it's very relevant. But ultimately, we just won't know until some of these non-hallucinogenic compounds advance into phase 2 studies. Yeah. It's all speculation. And part of the reason that we're so interested across all of these hypotheses is because, that's, as a sort of data-driven scientist who's very inherently skeptical, the data is just not out there on any particular class as being superior to the other. So we're quite interested in actually exploring the science and developing compounds that might be relevant across a number of these different mechanisms.
Notice that PSYLO is also looking at less hallucinogenic compounds. Would this be a microdosing approach? Along with this question, could you explain to our audience the difference between non-hallucinogenic psychoplastogens and microdosing?
Yeah, sure. So microdosing is this term that makes me a little bit uncomfortable as a scientist, but the general concept is that you can take lower doses of psychedelics. Typically, these are described as being, subperceptual, so a dose of psilocybin or mushrooms where you ideally are not subjectively aware of anything happening. But people claim to use these in a sort of regular way, maybe 3 times a week. There's a protocol that was sort of popularized by Jim Fadiman around, very low doses of mushrooms taken 3 times a week. And people do report benefits. It's anecdotal, it's obviously subject to a few biases because people know what they're taking and how they're dosing. But the idea is that these below threshold sort of doses of psilocybin might give you improvements in anxiety, depression, and other behaviors as well, even non-disease states. So, in Silicon Valley, probably starting around the mid-2010s, it was, hugely popular among software engineers to be microdosing for creative flow and for other things. We're seeing some really interesting trials right now. Like historically, there was not much very good data out there for that sort of approach in terms of treating any disease, but people are now starting to do these trials. So one of our sort of colleagues at PSYLO is a guy named Vince Polito. He's at Macquarie University in Australia. He is looking at a very low dose, 5 milligrams of psilocybin in moderately depressed young males dosed 3 times a week. So you're sort of at this threshold where maybe you don't actually get any perceptual disturbances, but you may see improvements in depression symptoms. There's several of these trials now reporting. I think My Medicine had a similar one with LSD, much lower doses than would be typical for ADHD. So people are starting to look at these sort of sub-perceptual dosing, let's call it microdosing approaches to treating a number of indications, which is all well and good, except for, especially as the clinical data emerges to support these hypotheses. It's not quite there yet, but I'm optimistic that we'll get some answers in the next few years. But there is one problem with that approach, and that is that many of these psychedelics, almost all of the natural psychedelics, also interact with a receptor that's very closely related to 5-HT2A called 5-HT2B. And this is a bit of a problem because there was a combination drug actually called fen-phen a number of years ago that contained phentermine, a stimulant, and fenfluramine, which was an appetite suppressant. And fenfluramine's metabolite, although fenfluramine didn't target 5-HT2B too prominently, its main metabolite did. And in people taking this drug for very short periods of time, they'd end up with sort of pathology of the heart, valvular heart disease, and they'd end up dying of that. So the drug got a black box warning, and then as epidemiologists figured out what was going on, it was eventually withdrawn from the market. So the method by which psychedelics are used in assisted psychotherapy, this is not an issue because they're used very occasionally. 1 to 3 treatment sessions infrequently is probably not an issue. It's this chronic activation of 2B. So this is a real risk for microdosing because we don't know what that threshold is. How much 5-HT2B activation is a problem. So this is one of the biggest risks. So as we think about our less hallucinogenic series, this is a compound that in animal models suggests that it's not as hallucinogenic as it should be, given its potency for activating 2A. But we've also engineered it so that it doesn't hit 5-HT2B or other cardiotoxic liabilities. So it would be suitable should the clinical data support microdosing for a number of things. This is a class of compounds that would be suitable for that sort of application.
Is this also based off of tryptamine?
It is not based off of tryptamine, no. So within our library, I can tell you that the only compounds that are tryptamines are within the PSYLO 300X series. Everything else is a non-tryptamine scaffold.
Oh, wow. Okay.
So can you speak more about the kind of the scaffolds there, or is that?
That's fairly confidential. The general approach that we've taken, it's probably a good chance to discuss some of the computational approaches here. So one of the things we were very interested early on, I'd done some work with Brian Kabilka at Stanford and his lab, thinking about the relationship between a molecule structure and the way that it activates a target protein. So he won the Nobel Prize for methods to sort of characterize G protein coupled receptor function. This is, the largest class of sort of, therapeutic targets, especially for psychiatric medications. 5-HT2A is a GPCR. His lab had done a lot of work on mu opioid receptors, the sort of target of a number of different opioid drugs, CB1, the target of THC and cannabis and other things. And the approach here is that you can maybe take 5-HT2A, take its protein structure that's been experimentally determined, and there are a few different ways to do this, and now several of these structures have been published. You can take that three-dimensional structure, port it into a computer, start thinking about the place where psychedelics would bind and how they activate the protein in various ways. And instead of starting with the compounds that nature's provided or that various sort of scientists have provided over the last 50 years, we can actually take entirely new bits of chemical matter, both real and proposed, and start seeing how those molecules will interact with the protein. So we were doing this very early on, and we were modifying the 5-HT2A structures that were available with the experimental data from our lab to sort of refine that binding pocket. Ultimately, it led to the development of this platform that we've called PSYLO and trademarked that. And this is essentially a refined 5-HT2A protein structure, as well as several other proteins, where we can take very large sets of virtual chemical libraries and identify entirely new starting points for drug development from those. So 500X was its hits were derived from that approach. And in the case of 100X and 400X and a number of different scaffolds, we'd also use this platform to sort of optimize non-tryptamine type chemical structures for engagement with the protein.
Mm.
Understood. So I just want to kind of sum up what we were talking about in regards to 400X. So it will have the same, or you're aiming for the same efficacy as a psychedelic that would deliver a full psychedelic experience, but it's going to be less hallucinogenic, correct?
Based on the animal data that we have, yeah, it's a little bit tricky because this is not a compound that anyone's ever tried. No humans tried it. We don't do any sort of self-experimentation at PSYLO. We're not that sort of company. So it's a completely new bit of chemical matter, and we don't really know, based on its results in this head twitch response assay that I'm going to guess some of your audience is familiar with. It has a profile that is quite distinct from classical psychedelics like psilocybin, but has some sort of head twitch readout. So it's definitely different to 100X, and it's also different to our compounds from 300X. So we're terming it less hallucinogenic because it does potently activate the 2A receptor, but it seems to have a head twitch response profile that's somewhere between those two classes. So yeah, it's very hard to say in a human what its subjective effects would be, but based on the animal data, it should be distinct from 100X and 400 and 300X. Sorry.
That's actually really interesting. So for something like this, would you also be aiming for a shorter duration?
That's a great question, and we're not sufficiently advanced that we can probably answer that. Based on the sort of microdosing protocols, if we assume that it's doing some of the same things that are hypothesized to be occurring pharmacologically with the microdosing regimen of an existing psychedelic, it would probably be dosed fairly similar to that, which is actually probably quite similar to what others are considering for these non-hallucinogenic compounds and our own 100X. These are probably not drugs that you need to take every day. But they're not going to be drugs that you take 1 to 3 times only in the course of a year, like with assisted psychotherapy. So it's very likely they'll be dosed, probably less frequently than once daily, but 1 or more times per week. We just don't have enough data to support the precise dosing regimen yet for these sorts of compounds.
So Sam, just going to leave you off with one question here to wrap this all up. So there are now like, there's many companies now targeting the 5-HT2A receptor. I feel like every week or two I hear of a new company, even Intracellular Therapeutics, I think is getting involved now too. $6 billion market cap company was focused on, I guess, maybe more traditional psychiatric medications. And I thought that was interesting. But so a lot of companies now are interested in this receptor target. I'd like to ask you, how does PSYLO plan to differentiate themselves from other companies in the space looking at this receptor target as well?
Yeah, differentiation's sort of always front of mind for us. The intracellular story is an interesting one. I know a little bit about that if your audience is interested. So they developed a drug called Caplyta, lumateperone. It's a very well-known antipsychotic. And antipsychotics of that, atypical antipsychotics, are traditionally 5-HT2A antagonists. So this was a drug that was better than other antipsychotics because it had less sort of off-target activity at some sedating receptors and other problematic targets of historical compounds. So it was this sort of next-generation antipsychotic. And then a couple of years ago, you might have seen the paper, I can pull it up. There was a Chinese lab who actually used the sort of tetracyclic core of that drug, lumateperone, to show that you can control, by controlling the 2A's receptors dynamics, you can get compounds that either do or do not have this sort of hallucinogenic-like properties in mice. So I suspect that someone at Intracellular has seen that this scaffold was used in this sort of key paper coming out of China, and they already have an enormous sort of library of various related compounds, and they've seen the interest in the space and have just decided they can quickly pivot into development in this area too. Wow. So yes, I saw a poster on that compound at a recent scientific conference, and yeah, I was kind of shocked to see it as well, but it actually makes a lot of sense if you think about the historical development. So in terms of differentiation, I guess one of the main things about 5-HT2A is that of all the GPCRs that we know that are very well characterized, there are a lot of them.
Sam, can I just interrupt here? What are GPCRs?
Oh, sorry. Yeah. So GPCRs are a very important class of therapeutic proteins in the human body. It stands for G protein coupled receptor. And you can think of them as sort of like a cellular switch of sorts. So a drug, or in the endogenous sense, so in the sort of native sense, a molecule that we have in our body comes along, targets one of these GPCRs that it's designed for, is evolved for, and then induces a protein on the other side, this G protein complex, to dissociate and go off and do a whole bunch of different activities. So it's sort of a way for a cell to signal from one side to the other, a cascade of activity. So our body's got hundreds of these things, these different types of receptors. Many of the drugs that you'd be familiar with that have been developed over the last couple of hundred years target GPCRs. They're a very popular drug target. We've had good success at developing drugs to target them. The drugs typically come in two forms. So you have drugs that activate these things or block them, so agonists or antagonists. Okay. There are now more subtle sort of developments. So there are things like allosteric modulators that just sort of fine-tune responses to the endogenous transmitters. So this is a constantly evolving field of science. And you've got companies like Septerna now looking at how to modify other GPCRs that were considered undruggable or that we don't know much about. So, a very constantly evolving, super interesting area of scientific research. What's notable is that historically 5-HT2A, despite being the main target for a lot of antipsychotics, in the antagonist mode, drugs that sort of block this site, very few drugs were ever developed to activate 5-HT2A because the dogma was that if you activate this receptor, you'll get hallucinations. And for many compounds, that's true. Certainly, we've got plenty of examples from the tryptamine space, the ergoline space, things like LSD, phenethylamines, DOI, and other drugs. But the reality is that the science has now advanced such to the point that we know we can rationally engineer compounds that can activate the receptor but don't cause hallucinations. And there's a few of these examples starting to appear now from companies like ours, from Delex and others. So the approach, the idea of targeting 5-HT2A and activating it, I think is quite a new idea. And it's sort of like a big gap in modern drug development because of this historical misconception that it would always cause hallucinations and therefore is a bit of a liability. So we're sort of catching up on 70 years of research that didn't happen because of preconceptions around this protein as a target and the taboo around sort of psychedelics as areas for development. So most companies sort of have a different idea about how this protein can be targeted. So companies like Delex are pretty focused on the hypothesis that various substances, targeting 2A and even other receptors in different specific sites of a cell might lead to neuroplasticity, and that's fundamentally what drives a lot of this. I believe Gilgamesh probably has a different hypothesis. Most of their compounds seem to be psychoactive in some way. And some people believe that, you need to activate 2A, but you do need those hallucinogenic effects as well. So we're a little bit unusual in that we have hedged our sort of pipeline across both hallucinogenic and non-hallucinogenic compounds because, we're still developing the science. But we are pretty heavily focused on 2A and combinations of 2A with other receptors where we think there might be a synergistic utility there.
Okay.
So our efforts are really largely focused on 2A because we believe this probably is the most important target, either alone or in combination with other proteins, where it forms dimers that is driving a lot of the therapeutic activity based on activation and a lot of other downstream signaling.
Awesome. It's very interesting. It's always fascinating to follow companies like yours working on, these truly novel, psychedelic inspired, medicines. And I can't wait to follow the rest of the work you're doing in PSYLO. And to our viewers, you should check out PSYLO's newsletter. It's really incredible. It breaks down the science of psychedelics in a way that's pretty easy to understand, which is really much needed in this space. And Sam, would like to thank you so much for coming on today. Adam, thank you. And with that, if you guys enjoyed this video, enjoyed this content, please like and subscribe. Check out our new website, psychedelicvantage.com, and we hope you have a great day.